So, welcome again. Now, we are starting our third session and again we are on the secondary structure of protein and dealing with alpha helical pattern. So, these are the collagen fibers and in this diagram we can see tropocollagen molecules with 3 standard polypeptide chain units. They are arranged head to tail in the parallel bundle. Each unit of tropocollagen is about 1.5 nanometer wide and 300 nanometer long. Series of complex covalent crosslinks are formed within between the tropocollagen, makes it strong mature collagen, and this is the reason for rigid brittle connective tissues in old people. Now, homotrimers, if there are more than one polypeptide chain forming a particular structure, this amino acid chain can be homotrimers. That means, if all the amino acid residue in a sequence they are identical, and it can also form as heterotrimers if the two chains are identical and the third ones differ. So, all the three chains they can be identical or it can be differ. So, it can comes to homotrimers or heterotrimers, and in the diagram you can see the electron micrograph of collagen from skin. Now, glycine and proline, this bulky amino acid, charged amino acid favor disruption of helix. The disruption of helix is important as it introduces additional folding to the polypeptide backbone to allow to form a globular protein. Now, genetic disorder of this collagen. Now, we know that. So, this shows the close relationship between amino acid sequences and the 3D structure of protein. Two main disorders are seen here. We can see the characters which are characterized by loose joints. A group of 10 different collagen deficiency in this disease causes fragility, hyperdensity of skin, hyperextensibility of the skin caused by defect of type 3 collagen. So, arises between glycine and is replaced by serine. So, it is a genetic disorder, you can see, because glycine is replaced by serine. So, it is characterized by loose joints and collagen deficiency diseases, fragility and hyperextensibility of the skin. So, these are caused by the collagen deficiency type 3 collagen, and it is a genetic disorder. Here you can also see the hyperextensibility, you can see here. So, it is ED syndrome, the Indian rubber man we can say. Then also we can see it is also known as brittle bone diseases, abnormal fragile bone formation in human babies arises because of glycine is replaced by cysteine. So, we can see the primary structure of protein is very important. Any mutation it causes the replacement of amino acids, and if the amino acid is replaced by another amino acid, the property is totally changed. So, mutation which can this mutation it causes deformity and the genetic disorders. So, in this case glycine is replaced by cysteine, and defect is the synthesis of type 1 collagen. In this case here the defect is in synthesis of type 1 collagen. So, numerous fractures and severe bone deformity, small stature with underdeveloped lungs we can see here. These are the all type of deformities which are seen only by the replacement of glycine by cysteine. Then here you can see the different type of babies formed by such type of mutations, genetic mutation, replacement of amino acid which causes the deformity in the structure, in the bone, in the overall body stature. Now, coming to beta pleated sheet. Beta plate is secondary structure of protein. So, beta pleated sheets are composed of 2 or more different regions of stretches, at least 5 to 10 amino acids. The folding and alignment of stretches of the polypeptide backbone aside one another to form beta sheets is stabilized by hydrogen bonding between amide hydrogen and carbonyl hydrogen oxygen. So, there are what is the difference between a beta pleated and alpha helical structure? Here the hydrogen bond formation in alpha helix is in the same intra chain in the same polypeptide chain, whereas in beta pleated sheet it is stabilized by two different chain, polypeptide chain. So, in the side direction, unlike the compact backbone of alpha helix, the peptide bond of beta pleated sheet is highly extended in the form of a sheet. Beta sheet are said to be pleated in which the R group of adjacent residues point in opposite direction. So, this is the orientation of R group which are pointed in opposite direction. Now, the arrangement of beta, because the hydrogen bond formation is between two different peptide bonds. So, arrangement is also seen there. So, beta pleated sheet, the hydrogen bonds can be between a parallel polypeptide or in anti-parallel sheets. So, beta pleated sheets are either parallel or anti-parallel. Now, here we can see the hydrogen bond formation between the two different polypeptide chain laying side by side. So, this is beta pleated sheet where two different polypeptide chain, the hydrogen bonds is formed between two different polypeptide chain, unlike alpha helix structure where the hydrogen bond is formed between the same polypeptide chain, intra chain formation. Now, it was also identified by Pauling and Corey. Formation depends on intramolecular hydrogen bonds, which is formed by parallel alignment of number of polypeptide chains. Individual polypeptide chain, that is the beta strand, they are stabilized by hydrogen bonds between NH and carbonyl group of adjacent chain. Now, you can see here both the structure, alpha helical structure and the beta pleated sheet. In parallel sheets, adjacent polypeptide chain proceed in the same direction, that is direction of N terminal and C terminal ends is the same, we call it as parallel. In parallel sheets, because the conformation is both parallel and anti-parallel. So, what is about parallel sheet? Parallel sheet means the adjacent polypeptide chain in which there is between where the hydrogen bond is formed, it is in the same direction, that is direction of both the polypeptide chain of N terminal and the C terminal is the same. Whereas, in anti-parallel sheet the adjacent chain are aligned in opposite direction. So, say if the one polypeptide chain is having N terminal at one end, the second polypeptide chain is having C terminal at the particular end. This is the secondary structure of protein showing alpha helical and beta pleated sheet. The beta pleated sheet is showing in the parallel direction, the N terminal and the C terminal of the adjacent polypeptide chain are in same direction. It is local regularly occurring structure in the proteins. So, we can see the hydrogen bond formation in alpha helical and beta pleated sheet. Now, there are also seen random coiling. It is also known as the third type of secondary structure of protein, which is called as the random coiling, where a polypeptide chain contains adjacent bully residue or the charged residue. Repulsion between these two groups causes polypeptide to assume a random coil configuration. So, it lacks a well defined structure. In spite of that, there are certain other secondary structures such as B turn and collagen helix. Now, this is the beta turn, also known as beta blend or hairpin bend. It is found in the surface of protein and also called reverse turn. You can see proline and glycine and their formation, and there is formation of U turn, or we can call it a beta turn. It is a hairpin like structure, therefore, it is known as hairpin bend. It permits the change of the direction of polypeptide chain to get a folder structure. It gives a protein globularity rather than a linearity, because this structure beta pleated sheet, they are linear in structure, but due to this bend the protein get a globular structure. Hydrogen bond stabilizes the beta bend structure. So, where it is bent, the hydrogen bond, it is the hydrogen bond which is stabilizing the structure. The proline and the glycine are frequently found in beta turns. So, definitely we have seen in the diagram that it is the proline and the glycine residues which are actually responsible for the bend of these beta structures. Beta turns often promotes the formation of anti-parallel beta sheets, occur at protein surfaces. It involves 4 successive amino acid residues. So, irregular secondary, we can also see irregular secondary structures, non-repeating loops and turns, and then we can see the change of directions, and this change, as we have seen, it turns, it actually have about 4 amino acid residues, and internally they are stabilized by hydrogen bonds, and in this turn we can see the amino acid glycine and proline. Now, here we can see the beta pleated sheet, parallel sheet, anti-parallel sheet and the mixed sheet. So, we can see beta pleated sheet in all the three forms, either they are parallel having the same amino terminal group at the same end and carboxy terminal and the same end, or anti-parallel opposite direction, or mixed one. Now, this is the beta pleated structure. It is found when two or more polypeptides line up side by side. You can see both of the polypeptide chain, they are lined side by side. Individual polypeptide chain, there are 10 beta strand. Each beta strand is fully extended sheet, it is in the form of a sheet, and they are stabilized by hydrogen bonds between N and NH and carbonyl group of adjacent chain, hydrogen bonds between the two adjacent group. This is the basics of formation of beta pleated sheet. Now, here you can see the anti-parallel sheet. In the first diagram you can see the anti-parallel sheet where the amino terminal and the carboxy terminal, they are in the opposite direction and they are forming a loop, hairpin loop like structure, whereas in the second diagram we can see all the three polypeptide chain, they are having the amino terminal end at the same direction and carboxy terminal end in the same direction. So, you can see a big loop, it is coming from amino terminal to carboxy terminal. So, we were talking about psi, pi and omega and interpretation of these bonds and what is the relation of Ramachandran plots with it. So, you can see the Ramachandran plots, how it is plotted and how it shows the angle between pi, psi and omega angles. So, you can see the white region, the red region and the yellow region. White region, it shows sterically is allowed for the amino acid except glycine, and it is the red region, it allows region, namely that is the alpha helical and beta pleated sheet conformation, and the yellow areas, they are the outer limit. So, this is how the Ramachandran plots it is interpreted, how the angle psi angle, pi angle and the omega angle. Now, the uses of Ramachandran plots, it can be used in two somewhat different ways. First, to show in theory which values of conformation of psi and pi angles are possible of an amino acid residues in a protein, and second is to show the empirical distribution of data points observed in a single structure. So, therefore, it has a very significant value. Now, the protein foldings, what are actually these protein foldings? The peptide bonds which are a linear sequence of amino acid, they allows for rotation around it and therefore, the protein can fold and orient its groups in favorable positions. So, weak non-covalent interaction which holds the protein in its functional shape, these are the weak and will make many of these holes of the shape. So, that is why the protein it gets fold, and we can see a many and it is very significant why a protein folds, where it folds and how it folds. After the linear sequence of an amino acid or the primary structure of the protein, which is a linear structure, the protein fold in a very specific manner and according to their function, and this protein folding occurs in the cytosol. And because the peptide bond that allows for the rotation around, and therefore, the protein can fold and orient its group in favorable position, and actually which decides or which implicates its function of the protein. And for this protein folding, weaker non-covalent interactions are responsible and which gives protein a functional shape. So, here we can see two regular proteins foldings pattern which we have seen here, loops, bonds of peptide backbones, alpha helical, beta pleated and also the irregular folding. Now, turns and loop which we were talking about, proline and glycine, they form a turn and loop which is stabilized by a hydrogen bond. These are the beta turn loops we have already discussed earlier, more than one fold. Now, protein folding is also a very significant value. Traditionally, one protein can fold one Now, these are the different type of proteins which are folded. Metamorphic proteins, they fold. Now, sometimes when the proteins they are not folded or the proteins are midfield, the proteins they cannot perform their function what we are assigned to, and we can see the hereditary or the genetic diseases. These are the misfolding. The misfolding of proteins, it leads to pathology. Now, we can see the Parkinson disease, the Alzheimer disease, the prions, and there are so many diseases and genetic diseases which are related with misfolding of proteins. So, protein, now we can see each and every part is very important. The sequence of particular amino acid is a primary chain, then this primary structure, how it folds in the cytosol, and if there is a mutation and if an amino acid gets replaced by another amino acid, you can see the folding it can be also affected, and the folding of proteins definitely affects the function of that protein and which will lead to pathology. Now, there are certain motifs. What are motifs? Motifs are recognizable combination of helix, loops and sheets. So, these are also super secondary structure of protein. We have already discussed the loops, the motifs which have helix turn helix. We can see helix turn helix loops, it is seen in the diagram, then helix bundle loop, then hairpin loop, beta sandwich loop, all these loops it is shown in the diagram. Now, there are some other motifs which are highly studied. Some of them are leucine zipper, zinc figure, and often there are so many applications of this. These are different type of motifs which are found in the super secondary structure of protein. Again, secondary structure of the protein often constitute distinct domains. Now, what are these domains? Domains is the basic unit of structure and function. Now, these are the domains. Domain is a basic structural unit of the protein structure distinct from those that made up of conformation. So, conformation are different things and domains they are different things. They are the part of protein that can fold into a stable structure. Different domains can impart different function to proteins. So, domains are the functional part of the protein, and these domains they have their different functions. Protein can have one to many domains depending upon the protein size. What is the size of the protein? So, one protein can have more than one domain. So, it also signifies its function. The tertiary structure of protein describes the relationship of different domains to one another within a protein, or the final arrangement of domains in a polypeptide chain. The interaction of different domains is governed by different forces. So, how these domains are formed and how it is related to the function of protein and what leads to the formation of different type of domains in a protein. So, there are different forces. These forces are hydrogen bonds, hydrophobic interactions, electrostatic bonds and van der Waals forces, which gives domain its specific structure to its function which is it has been assigned. Alpha helical and beta sheet, the hydrogen bonds, this eliminates the possibility of water to interrupt the structure of protein. This is its basic structure, alpha helix and beta sheet. The hydrogen bond, this eliminates the possibility of water to disrupt the structure of proteins. Now, polypeptide chain which have more than 200 amino acids in length generally consist of 2 or more domains. We were talking about that a protein can contain one or more domain if the protein is of higher molecular weights. So, in such case if the protein is made up of more than 200 amino acids, then we can see 2 or more domains there, and the core of domain is built from motifs. So, we have also seen the motifs in the previous slides. Now, the folding of protein chain within the domain usually occurs independently of folding of other. That means, if there are 2 domains in a single protein, so folding of one domain is independent of folding of another domain. That means, both of the domains, the folding of both of the domains, they are independent, they do not disturb or affect each other. So, each domain has a characteristic of a small compact globular protein. It is independent of other domains. So, both of the domains, the folding, the characteristic feature, they are independent. They are not dependent on each other, or they do not affect the folding of one domain does not affect the folding of another domain. Now, these are the structural and functional domains. They are discrete, they are independently folded units. So, each domain is a single unit. They maintain the shape when cleaved or the loop. So, it is also a very interesting character. The domain, they maintain their shape when cleaved. They may have separate activities. Domains, each domain had its separate activity. Separate activity means separate function. So, ATP binding domain or catalytic. Suppose if the protein is having two of the domains, one domain may be linked with binding of ATP, adenosine triphosphate, and another domain it may be have the binding of a substrate, that means the catalytic domain. So, similar activity, similar structure, it may also occur across many proteins. Binding pockets. So, these are the binding pockets or at the interface. So, it may have a regulatory, the domains, they may have the regulatory function or the catalytic function. So, these are the common domains which are seen, the structures of the common domains, it is seen here. So, this comes to the end of a secondary structure of protein. So, in this secondary structure we have seen alpha helical structure, beta pleated sheet, super secondary structure of proteins, domains, motifs, loops, and also some of the random folding, and also the misfolding, what happens if there is misfolding of proteins. So, till we have learned about the secondary structure of protein. Thank you.